मंदिर एक ज्ञान साधना, ज्ञान साधना.
विद्या मंदिर एक ज्ञान साधना, ज्ञान साधना.
ज्ञान साधना, ज्ञान साधना.
ज्ञान साधना, ज्ञान साधना.
जाती भेद होती लुप्त या स्थळा, प्रगतीचा मार्ग सुज्ञ दाविती जगा.
वर्ण जाती भेद होती लुप्त या स्थळा, प्रगतीचा मार्ग सुज्ञ दाविती जगा.
कला शास्त्र वाणी क्रीडा रुजवी मन मना, ज्ञान साधना.
विद्या मंदिर एक ज्ञान साधना, ज्ञान साधना.
लक्ष साधण्यास दिव्य दृष्टी लाभते, घेण्याहून देण्याची वृत्ती जागते.
लक्ष साधण्यास दिव्य दृष्टी लाभते, घेण्याहून देण्याची वृत्ती जागते.
स्वप्नपूर्तीचा उदात्त हेतू भावना, ज्ञान साधना.
विद्या मंदिर एक ज्ञान साधना, ज्ञान साधना.
पंखाना बळ लाभे या इथे सदा,
पंखाना बळ लाभे या इथे सदा.
बुद्धीचा सूर्य नवा उजळी अंतरा,
बुद्धीचा सूर्य नवा उजळी अंतरा.
यश कीर्ती भावी हाच ध्यास गुरुजना, ज्ञान साधना.
विद्या मंदिर एक ज्ञान साधना, ज्ञान साधना.
ज्ञान साधना, ज्ञान साधना.
ज्ञान साधना, ज्ञान साधना. ज्ञान साधना, ज्ञान साधना. ज्ञान साधना.
Hello everybody, hoping that you are safe and in good health. On behalf of the Satish Pradhan Jnana Sadhana College, Thane, Maharashtra, India, I, Dr. Sarita Hajirnis, Associate Professor and Head, Department of Botany, extends a warm welcome to all. The present pandemic situation with the frequent lockdowns has forced the education system to change the teaching and learning methods. E-learning now plays a major role in the new normal. Webinars are one such effective tools of sharing information and knowledge through internet and connecting people from distant places. Satish Pradhan Nyan Sadhana College, Thane, in collaboration with the Mycological Society of India, Mumbai Unit, have jointly organized today's webinar on the topic Rare Fungal Spores as Trace Evidence and Intelligence in Forensic Investigations. This will be presented by the reputed and eminent British mycologist and lichenologist, Professor David Leslie Hawksworth. I now invite Principal of our college, Dr. C.D. Marathe, to present his opening remarks.
Namaste. On behalf of Satish Pradhan Nyan Sadhana College, Thane, I welcome all the participants, members of the Mycological Society Mumbai Unit, and today's speaker, Honorable Professor Dr. David Leslie Hawksworth, to the one-day international webinar on rare fungal spores as trace evidence and intelligence in forensic investigations, arranged by the Department of Botany and Internal Quality Assurance Cell of the college in collaboration with Mycological Society of India, Mumbai Unit. To tell you in brief, ours is a co-educational institute established in 1980 in which more than 8,000 students are enrolled for various programs, undergraduate, postgraduate, and doctoral level. These programs range from arts, science, and commerce, including computer science, information technology, film, TV, and multimedia. I understand a little about fungi, and I am a complete layman. Very little information gathered reveals that fungi, as the common man perceives, are organisms responsible for spoilage of food, or that what grows on clothes, leather in damp weather. However, we want to understand that in reality, they are of tremendous importance to the common man through their applications in industry, pharmaceutical, medicines, bakery, waste degradation, and in research and academics. Today, we'll be enlightened on one valuable aspect of fungi, and that is forensic science. We are glad to welcome with pride Professor Dr. David Leslie Hawksworth as our guest speaker today. Sir, we are really privileged to have you today. We are also proud and delighted that there are 1,750 plus participants from 11 different countries apart from India. Without taking much time, I wish the organizing team best of luck, and I'm sure that the whole purpose of arranging this webinar will be definitely served. Thank you.
Good morning and welcome to the webinar series of MSI Mumbai Unit. The Mumbai Unit of the Mycological Society of India was established in the year 2005. The basic idea is to encourage the students and young researchers to get acquainted with the recent trends in mycological research and work on exploring the fungal diversity of Western Ghats, one of the hot spots of biodiversity. The other important area is to explore marine fungal diversity, along with this, the industrial applications. The decision on MSI Mumbai Unit was taken in the annual meeting of the Mycological Society of India in the year 2002, and the unit was established in 2005. Dr. R.G. Bagul was the first chairperson and Dr. Vikas Gulatkar as the secretary. Dr. M.C. Srinivasan from NCL Pune and Dr. S.K. Deshmukh from Piramal Enterprises Limited were the member of their advisory committee. We have organized the first webinar in the year 2005 at Tuya College, Mumbai. We are having an annual conference on various aspects of mycology. In these meetings, the presentations were made by all eminent mycologists from our country, including late Professor K. Natarajan, Professor Surnarayan, Professor Bhatt, Professor Manorachari, Professor Atri, and so on. It will be unfair if I do not acknowledge the support of late Professor K. Natarajan and other MSI office bearers. Speaker of the day, Professor D.L. Hawksworth is well-known mycologist and lichenologist who always supports the youngster to work on various aspects of fungi. I am thankful to him for readily accepting the invitation. I welcome him on behalf of the Mycological Society of India as a past president and president of Mumbai Unit. Professor Suryanarayan is the moderator for today's presentation. Now I will request Dr. Shilpa Verekar, Head Microbiology QC at Parle Agro, Mumbai, to give brief introduction of Professor D.L. Hawksworth. Over to Shilpa.
Good afternoon. The task of introducing our eminent speaker of today, Professor D.L. Hawksworth, is a great privilege. Professor D.L. Hawksworth is a CBE, Commander of the British Empire, which was appointed by Her Majesty Queen Elizabeth II for services to science in the year 1996. Sir is a PhD DSc from the Leicester University. Sir is the recipient of many medals and awards like the International Mycological Association in 2018, Founder Award of European Mycological Association Wiltshire 2015, Einswan Medal International Mycological Association Bangkok 2014. Sir is a registered environmental biologist, Institute of Biology in 1990. He is a chartered biologist in 1986. He is the Fellow of the Royal Society of Biology 1982, Fellow of the Linnean Society of London 1969, Fellow of the Royal Society of Advanced Arts, Manufacture and Commerce 1997, Professor Member Chartered Society of Forensic Science in 2011. He is also an honorary member of the Mycological Society of America, British Mycological Society, Centenary Fellow, the Latin American Mycological Association, British Lichen Society, Lichenological Society of Japan, and Italian Lichen Society. Sir, the Professor of Colorado Doctorate Universitat Complutense de Madrid 2001 to 2016. He was a Professor of Biology and Ecology at the University of Gloucestershire 2007 to 2009, 2013 to 2014. Council Member of English Nature from 1996 to 1999. Mycologist at the Commonwealth International Mycological Institute Kew 1969 to 81. Director of the International Mycological Institute Kew in Ingham 1983 to 97. Sir has held many academic positions. Some of them are, he was an Honorary Research Associate, Royal Botanical Garden Kews from 2014 till date, Scientific Associate, the Natural History Museum London 2006 till date. He was the President of International Union of Biological Sciences 1994 to 97. He was the President of International Mycological Association 1990 to 94. President of the British Mycological Society in the year 1990. President of the British Lichen Society 1986 to 87. He is also a visiting professor to many universities like the University of the Kent, London, Reading and Southampton, UK, Jilin Agriculture University, Changchun, China. He is the Editor-in-Chief of International Mycological Association from 2010 till date, Editor-in-Chief Biodiversity and Conservation from 2006 till date, Executive Editor, Senior Editor of Mycological Research 2000 to 2009, Book Review Editor of Mycotaxon from 2000 to 2009, Co-Editor of Systematic Ascomycetum 1986 to 97, Co-Editor of Editor, Coordinating Editor, Mycopathologia 1984 to 87. Sir was the Chair IUMS International Committee on Bionomenclature 1995 to 2012. His past teaching experience includes, he was teaching MSc students of fungal biotechnology at the University of the Kent, pure and applied and fungal taxonomy, medical microbiology, and so on. Sir has also been a guide for many of the MSc, PhD and DSc thesis. His research interests are basically fungal diversity, systematics and nomenclature, especially of Ascomycetes, lichen-forming fungi, and fungi that obligately occurs on lichen. Currently, he is working on the molecular phylogenetic approaches in resolving classification and utilizing fungi in forensic investigation and environmental surveys. Sir is also a consultant to many of the firms. Sir's publications include, he is the single author for 57 books, co-author of 600 refereed papers, which includes 7 items in Nature. These includes authorship in 892 new scientific names, including 15 orders, 12 families, and 79 genera new to science, 247 species new to science, and 488 new combination or new name to the end of 2016. His ResearchGate, he has 899 research items. His reads in ResearchGate are 1,62,259 with 26,904 citations. Sir has written numerous books. There is a big list. I will just cover the recent one, that is the International Code of Nomenclature for Algae, Fungi and Plants, which was in July 2017. It's an honor, sir, to have you here. I will invite sir to proceed.
Good morning, everybody. Can you hear me okay? Anyway, welcome. This is
Yes, sir, you're audible. Yeah.
Hello? Am I audible?
Yeah, sir, you're audible.
Okay, thank you. Okay. So I'm with my wife, Professor Patricia Wiltshire, who is very involved with the forensic side of things, and I'll say a little bit more about that as we get started. Thank you for those very kind words. Which I'm sure you've been rather looking at pictures to do with fungi rather than that. But anyway, I'm very flattered that you've organised this and have said so many kind things. Thank you. Shall we move on to the presentation as time is getting short?
Yeah, sir, we are loading it.
So this is concerning one aspect of the use of fungi in forensic work, where rare spores can give extra evidence to add on to the other information from pollen and so on. And I only became involved in this very late. Oh, next slide, please. Next. This was just a note. Some of the slides this are a bit sensitive or original, so we don't want any copies of this retained or made. Professor Wiltshire had a big issue some years ago where somebody videoed one of her presentations and was going around America giving her talks for large amounts of money, and so we're rather sensitive about this. Thank you. Most of the data is in some references that we'll give to you. Next. So the history of my involvement with this is that Professor Wiltshire was an expert in palynology and its applications in ecological history and latterly in forensics. Got me to identify some fungal spores in some murder cases and thought I'd be very useful. One of her colleagues thought that I should be cultivated, and so eventually we got married. But after I'd been tested out at dealing with dead bodies, people who recently been murdered in situ, and had to appear in court in the Old Bailey in London. You can read more about her fascinating life in this book Traces that came out last year, and the American version, The Nature of Life and Death, if you prefer American spellings. And it's also just been released in paperback, as you see at the bottom there. Next. Well, first of all, I wanted to try and say why fungi add such a useful dimension to other forensic investigations. And that's basically because there are so many of them. And so these give potential bits of trace evidence that could be added to other things there. And this is a review of the issues to do with numbers of species that was published initially online in 2016 and then in book form in the Fungal Kingdom in 2018. Next. So basically where we are today, if we assume there's just 2.2 million worldwide, and some people say there should be 12 million worldwide, that's another story. But we only have names for about 140,000 fungi, which means that about 2 million need to be described, or 6% are known. And the consequence of this is it's very difficult to necessarily name the fungi that you might find in any particular sample. Next. Many people try and do identification through molecular work, and this slide is really just for a warning. We may only know about 6% of the species that are on Earth, but if we look at the numbers where there are sequence data deposited in GenBank, that's just over, well, just under 35,000, and that's only 25% of those that have names already. So something like 94,000 fungi with names that are just not represented in the databases. So it's very dangerous just to name things using the databases, particularly also as ITS sequences can be identical in fungi that are actually different species in some cases. Next. And you can find these new fungi absolutely everywhere. This is a case where we were involved in surveying the organisms of all kinds, including fungi, in Buckingham Palace Gardens in London. And this attracted a lot of interest in the newspapers because 2 species new to science fungi were described from the gardens. And this was quite a surprise because people didn't think that. We find many more fungi, of course, in other places. Next slide. Next. Just to illustrate this, the Vestivik Fungal Biodiversity Institute in Utrecht, this is what used to be the Central Bureau for Schimmelculture, started a project getting children and other citizen scientists to collect soil samples, mainly around Amsterdam. They got 293 samples, and these represented 1,840 species, which they decided they would name after the collectors. But of these, they found 1 new family, 2 new genera, and 25 new species in the first year of this project. So very, very rich things can be found without going very far. And of course, when you come to situations like India, it's just amazing how much still needs to be discovered. Next. There's a great variety of fungi, of course, and some of these have very distinctive spores. That means you can recognize them in some cases from them. That's not true in all groups. Some have very similar spores in different species, but some have very distinctive ones. The next slide. These are just examples of some of the spores that we've used in forensic investigations to get added evidence to the other evidence that's been available. Next. And we've been able to use these spores to help a wide range of things, linking objects, people and places, as trace evidence, locating human remains and graves, differentiating crime scenes from deposition sites, finding origins of objects or materials, eliminating places as potential crime scenes, eliminating suspects, demonstrating false rapes and other errors, estimating time since body or other materials have been deposited, estimating post-mortem interval, and in some cases the cause of death. So forensic mycology can be used in all sorts of areas, and today we're really going to be looking at the trace evidence side, in particular linking objects, people and places. Next. Fungi can grow on all sorts of substrates, as you'll be well aware. Some of these are illustrated here, and this means that you can take samples from all sorts of things involved in cases and get things that you can actually analyze for the all the materials that are in those, including, of course, pollen, and particularly relevant today, any fungal spores that might be there. Next. Now, the methods that are used we found valuable are basically Professor Wiltshire does work on the pollen primarily, although she has lot of experience in mycology, and the protocols for getting the samples out of things are described in this paper in the journal Palynology 2015. And this describes how things are sampled, how extracts are made of pollen and spores, how samples are treated, and how they can be interpreted. And it's important to realize that it is actually, you need the basic palynology techniques that have all the pollen data. It gives a lot of information about sites. And then amongst these, we find some fungal spores that give added value to the data that comes from the pollen, a separate sort of evidence that can support the data from the pollen or challenge it, although so far everything has worked together. Thank you. Next slide. This just gives an example. Very small amounts can be collected from things. This is a shoe, and from the sole of the shoe, just with a scalpel blade, a very small amount of material can be taken, and this is processed through the procedures described in that paper, which involve using some rather nasty chemicals, particularly if there are silica grains and other things there that need to be dissolved with sulfuric acid. So it's quite a delicate process, and it does require access to a laboratory that has a fume cupboard which is resistant to hydrofluoric acid. But after treatment and staining, we end up with microscope slides. Again, next. It can be a bit of a muddle like this. Lots of different things are mixed in there. A lot of background material. As much as possible has been dissolved away. And you can see there is a big fungal spore in the middle, other fungal hyphae and things around. And in some cases, the spores can be very characteristic and give us a lot of information. But also in all these samples, the pollen and any other small organism, other what we call palynomorphs, any small structures, may be relevant as well. Next. I just want to give you some examples now of particular fungi that turned up and why they were interesting. This is one called Caryopsera calicarpa, an ascomycete. And there was a case where a body with its head and arms, hands cut off, was found by the side of a stream. And the police wanted to know how the body actually got there. And this particular spore was found in the mud on the body and on the track on the other side of the stream. So it seemed very likely that the body had actually been dragged from one side of the stream where there was a track through the stream and dumped on the other side of that. Now this is important for finding the access routes and how the criminals might have got there. But this was very interesting. We mentioned rare spores in the title, and this was one that had bothered Professor Wiltshire for a long time, and she just had a drawing of it on her desk, worrying about what it might be. And I said, I know what that is, because I'd actually worked on this genus, describing another new species in it many years ago. And it was really quite remarkable because this occurs on rotten wood. But when we actually looked into the records, it had not actually been collected since 1865 by any mycologists in Britain. It is obviously around and has turned up in a number of archaeological sites and in forensic investigations. I've tried to get the amateur mycologists in Britain, who are very keen field workers, to look for it. I included pictures of it and so on in their amateur journal. But so far, there's still no fresh collection since 1865, although the spores are around and have turned up on a number of occasions. Next. An interesting case where a lot of work was done through the pollen, and I'll show a slide relevant to that later, to do with badgers. Meles is the name for the badger. And this is a protected species in Britain, but some people actually like to go out and kill them. They send dogs down their sets where they live to try and chase them out and tear them apart. The dogs often get killed, and the badgers come out and they will can get shot or whatever. Anyway, some interesting spores were found on a spade that came from this particular a person that was suspected of being involved in this, and these proved to be of a particular truffle. There are only 19 records of this since 2000 known in Britain, so it's quite a scarce truffle. Look at the next slide. And Patricia took samples in different areas to see whether they were unique or not, and identified that it was from the pollen data that it was in this area alongside this hedge. And what seems to have happened is that the badgers like collecting and eating truffles just like humans, and that they managed to get the spores of this thing in the samples from the set and on the spade, which indicated that the spade was used in this particular illegal exercise, and the suspects were convicted. And I think this was the first case, Patricia, wasn't it, for Royal Society of Protection of Cruelty to Animals to have somebody brought to court for this crime. These are very nice animals, because we have some that come in our garden, and we had a mummy and daddy and 2 baby ones last night feeding. Next. Another case that was interesting is that we were tested to see if we could say where 2 little bits of fabric that looked as though they were trousers had actually come from in the world. And Professor Wiltshire managed to get 639 pollen grains and plant spores from these materials, and 7 fungal spores. Well, the pollen data gave a lot of intelligence and indicated part of the area in the Far East or Eastern North America from the climatic requirements of the plants that were represented by the pollen. But the 7 fungal spores included this one, which was particularly pertinent, because this species, quite a lot of curvularia species, but I happened to have the monograph of these, and it transpired that the one that she got was one that's particularly frequent in wheat growing areas and in North America. And so it seemed that this had almost certainly been in Eastern North America, and when we were actually told where the samples had been, that was actually correct. So the fungi give added value data, and we're able to separate things between continents in that case. Next. This is one of the first cases I got involved in, where there was involving drugs, where a person had actually crept out behind an oak tree by a big cupressus hedge between it, and shot a person who had been dealing in a car in front of his wife and child. A very sad situation. And what was interesting is that this particular fungus is a mild pathogen of stressed cupressoid trees and shrubs, and it was a matter of trying to link the crime scene with the materials that this person actually materially had on him, a hood and so on, and also spores and so on recovered from the car. And what this was actually on the person, and it was very clear that he'd come from behind the tree, brushed against this hedge and picked up these spores.
I scrubbed the tree.
Yes, and Professor Wiltshire scrubbed the tree to get the spores from the tree to show they were on the tree as well. As a result of that, this was the first time that I ended up in a criminal court. There were actually 4 gang members prosecuted and convicted for involvement in this murder. So it's just icing on the cake, if you like. It's extra evidence which gives more support for a particular theory as to what happened. Next. This was an interesting one that really shows the power of the technique. A young girl claimed she was raped by a boyfriend. The boyfriend, the suspect, saying they'd had sex by agreement in a park at night. The girl claimed that he raped her amongst some trees and bushes in another place not very far away. Next. This just illustrates how close these places were. C is the park where the boy said that this had taken place. the crime scene with the sample A and B at the top, where she said it had occurred, and the girl lived very close. So you're dealing with places really quite close together, 100 meters, 130 meters apart. And one of the most powerful things with pollen data in forensic science is it can differentiate places remarkably close together, even, for example, things like different sides of a person's garden. But in this case, the pollen data was pretty good, but the fungal data was quite spectacular. Next slide. If you look here, there's material that came from the clothes of the suspect, the clothes of the victim, and things from samples from the wooded area and the park area. And you see there's a very close match between the fungi that are recorded and the suspect and the victim and the wood, and very little with the park. It's interesting with the one with the park, with Epicoccum nigrum, because this likes grass cuttings, dead grass cuttings, and the park had a lawn area, which you saw in that first slide there of this series, where obviously it was mown, and obviously had been grass cuttings there that this liked. So the fact they hadn't got much more of that, again, supported this. And it is important to stress that this is a lot of very painstaking work. And in this one, Professor Wiltshire and a colleague that helps account spores and pollen grains, they looked at counted over 38,500 spores. So the results might look quite interesting, but it is actually very hard work. The other thing is you never quite know what turns up in these samples. There's another rare fungus in there we thought was very rare, which is Diporotheca webbii, we called it. Next one. Next. And this was interesting because there was a Diporotheca rhizophila that appeared in some of the forensic literature. And I didn't really believe that this was the right fungus for the one that they've been using this name for, because I'd examined the original collections of that from America a long time ago, and there were no records of it outside part of North America. But when we actually looked at how the spores developed, it was quite clear they could be accommodated in the same genus, but it was a new species. This again is a case where you find something that's really quite special. We still don't know where this occurs in nature. It turns up often in remnants of areas where historically there have been swampy areas with trees, particularly older trees, but we don't really know. So you can get some very unusual spores that can help a lot. And this was described just based on the spores, and some other species of fossil species have been described since. Next. This was an interesting case where an old couple had been attacked with a hammer and things stolen from their rather isolated farm situation where they processed wood. You can see old bales of wood out there. They collected wood from parks and other areas and chopped it up and sold it basically. And there was a very interesting fungus on this that helped link the two suspects with the crime scene. It's a strange little spore with rather sort of a network on the surface, but it looks like little points depending on the optical view. Which again I happen to know because I'd worked on that particular group back in the 1970s. And this was interesting because I was able to show that there was a strong link between the escape vehicles. This turned up in two different vehicles and on the boots of the two suspects. So it was very likely they'd been at the crime scene and they actually confessed when confronted with the data. Next. This was a really quite fascinating and unexpected one. This was a bag that had been hidden and buried in a woodland in Northern Ireland, actually. And the police wanted to know where it had come from. It had guns in this. And one of the things that came there, so the pollen data was able to show what sort of woodland it had been in, which was really rather helpful. But what was surprising is that these spores turned up which is Thermomyces lanuginosus. Now, the minimum temperature for the growth of this fungus is about 30 degrees. It only really does well at 47 to 52 centigrade. And you don't get those sort of temperatures in a woodland in Ireland. And there are also a couple of unusual things there. A chetomium, close to a chetomium that was only known from deserts in North Africa. A Mixotrichotara, which likes to live on cellulosic materials, but usually in that nice warm buildings. So it's highly likely that this bag at one time had been in contact with a hot desert soil, buried in a hot desert soil or something. And it did actually transpire that they, from numbers that were on the guns that were eventually tracked down, these actually come from Libya. So it's really quite surprising how this helped and tied in. The pollen data helped show where the bag had actually been, and this helped indicate something about the bag's history and the guns. Next. This was quite a remarkable case where a colleague in New Zealand was trying to find out what these strange spores were in a sample of associated remains of a bird in a syrup that had come from New Zealand. You probably know there's a lot of interest and people interested in health buying Manuka honey. Well, it's obviously easier to make some surreptitious things, import some syrup and then sell it from New Zealand, and people might think it's Manuka honey. Anyway, they wanted to know where this syrup had come from, and I had a bit of a shock. The smut fungus is a smut fungus. They're very well known, actually, and there's some very nice monographs of these that have been published. And so I was able to find out what the spores were. And this fungus had been discovered in Brazil in 1959. Then it was imported into Argentina for study. But there were actually no modern records since I think about 1962, when they did experiments on it in Argentina, of this thing at all. But then I started to look into the plant pathology literature as opposed to the literature to do with smut identification. And there was a whole flurry of papers that since 2010 it arose causing problems in the groundnuts, peanuts, in the Cordoba area of Argentina, which is just where they'd done the experiments with this fungus in the early 60s. So in this case, there are no other reports from anywhere else. So in this case, there was a good indication that this syrup had come from South America, but most likely this is the Cordoba area of Argentina. So we're able to get really very good intelligence of where this might have come from, which really quite amazed us. Next. This was another smut spore, which smuts are very good for this sort of thing because they are very particular in what they grow on. And there was an issue of where a body had been deposited in Scotland, and this fungus, as this smut turned up, it had only previously been reported in Scotland in 1943 and 1999, and not subsequently. And it's associated with Glyceria and Phalaris grasses. And this is a habitat that didn't occur in the area they were looking at. And Professor Wiltshire was hunting around and looking at likely sites. And what she did find eventually was that it was a park which had a little pond, a small lake, a ditch that had these plants in it, which you wouldn't normally have expected. So that this was taken to be the place where a body had actually been deposited, although it would had the body had actually gone by that time. It had been devoured by badgers and foxes, but the spores linked the person's clothes to this particular site. Next. I just want to say a little bit about how one makes progress with this. There are a lot of books around about identification of spores from air, of varying degrees of reliability, I must say. But what it is important, if you're trying to identify these things, it's not easy. And if you try and look at things that are just in the air, it's a very small number. In the UK, we have about 14,500 species of fungi recorded, and I don't know what the total number is of fungi recorded in the air. I doubt it's much more than 200. So a very small proportion of the fungi that are around will be in the air. So if you're trying to identify things, you mustn't rely just on the books like this that are produced to illustrate the air spore. Next. The most useful thing in the temperate areas for things on plants is this microfungi on land plants produced by a colleague of mine, and his wife, who sadly now died, which gives lots and lots of illustrations of spores on particular plants, which is very valuable indeed. It is a matter of getting to know what literature is useful and where to look for things. Next. And there are databases that help if you do manage to identify a fungus or want to know what might be associated with a particular plant. There's a book that was produced in '89 by the American Phytopathological Society, which is also available as a database through the US Department of Agriculture group in Beltsville, which give a lot of intelligence about what fungus host combinations there are. So if you find a fungus which isn't confined to one plant, that may be on several, this is a good clue to things that it might also have been growing on. Next. In the UK, we're quite fortunate in that we do have a database that can be accessed online for the records in this country. Of course, it's not comprehensive because nobody can ever record every fungus everywhere it occurs, but it can give a very good indication of how common things are. Because if something only has 1 or 2 records, it's probably extremely rare. The ones down there is a Melanospora zamii. It was quite interesting in a case, the one that involved the Testudina, which I showed you some pictures of earlier, because the people that were processing the wood there also kept ducks and things around the back. And this is something, a fungus, which really is associated with maize rather than anything else. And this actually helped the case, because this was obviously associated with the decaying bird feed around where the criminals had been. In that case, the criminals did actually confess, if you remember from the slide. And getting confessions is very important because it saves everybody a lot of time and money. Next. Well, to identify things, the main thing is really to have a good reference library with lots of spores in and knowing where to look. And there's no easy way around this at the moment. It's a matter really of experience. And Professor Wiltshire thought I might be useful because I've been looking at fungi from or all around the world, particularly since the late 1960s, when I was appointed at the then Commonwealth International Mycological Institute, now no longer in existence. Because in the early days we used to have to identify fungi from anywhere in the world, and when you're a new mycologist on the staff, you had to deal with all the unidentified samples that came in. So you saw a vast range of things involved. And of course, over the years, we do lots of identifications, things you collect yourself in field work and things that people send to you. And so it's really just experience over the years that you need to know where to look to find things out. And then when you do find something, it's very important, if it is a bit unclear, that you go and check in the collections. Next. And at Kew, there's the largest reference collection of fungi anywhere in the world now. We're not sure how many species are represented, but there's probably near 1.4 million specimens there now. And the collections that were at my old institute are now at Kew, since that institute was closed. But not only are the reference specimens there, some have folders in red, as you see with the one that's pulled out there. And these are type specimens. These are the specimens to which the name actually belongs. Because when you name a fungus, what you're saying is that your fungus is actually the identical to this type specimen, the same species as that. So this is very important for verifying the accuracy of what you may have got. So next. So that's really where we are as value of using fungi as trace evidence and rare spores as train evidence. But the main thing for anybody involved in forensic work, if you come across any fungal spores you encounter, don't just throw them away or ignore them, because they may give key evidence or intelligence in a case. Next. These are papers that include some of the things I've been talking about. The one in bold, you can copy that slide with the greatest of pleasure. A couple of reviews that we've published. The first one, the cases there, is just about rarely reported fungal spores, with more information on those. And some of the other ones give evidence, give a background to how the fungal evidence was useful in some of the other cases we've described. So I'm trying to leave that for a minute, or you can send that to people later if you like. And next. So if you want to read something a bit more light-hearted than the science, buy Professor Wiltshire's book. I'm not sure how available it is in India. You can certainly buy it on the internet. You can get it through Amazon, and also you can, if you like to listen to things, headphones, it is available as downloads. What do you call them? Audio books. It's all audio books, which you can have either in American or in English. The American one is actually by Patricia. Next. So thank you very much, and then we come into question time. Hello?
Hi. Can you hear me?
Yes, indeed, yes.
Okay. So, Professor Hawksworth has shown us a completely new facet of fungi, working as policemen. It's a new facet, very, very enticing. So there are many questions. I quickly gathered all this and then sorted them out. A few of them were repeats, so I have now 5 questions to Professor Hawksworth. The first question is, in how many days sampling has to be done from the day of the crime to know the
Any time.
Well, the palynologist, I usually do the sampling. And what is wonderful about spores and pollen is they persist for such a long time. Now, with any crime, you should get there as quickly as possible. So if it's a murder, we should go very quickly while the corpse is still there, to give intelligence as well, because whole fungi can give you evidence as well. So the best thing to do is to go there when the body's there, as soon as possible to the crime, but the evidence can persist. So if you find fungal spores and pollen on someone's shoes, you can go back to the scene anytime and see if it's there.
Okay.
Yeah, question of the habitats as well.
Exactly. So that's very important. The basic fact here is some fungi are habitat specific. So we are able to zero down on saying that this spore comes from this place or this habitat. So we need to know more about fungi from different habitats. As David Hawksworth already mentioned in the first of his talk, we hardly know much about fungi. The second question is, what criteria are used to select the samples? This was the question, but it's too broad, I suppose. But anyway, it's for David.
Again, I will have to answer that for you. For selecting the sample, well, first of all, you have to work with what you've got. So if the police has just managed to get a pair of shoes or a car or a spade, or preferably all of them, you would work on all of them. But you have to relate that back to the crime scene. So at the crime scene, you really want samples from the offender footpath, from the way in that the offender accessed the site, all around the site itself. But very importantly, you have to have elimination samples, so that in many cases, a criminal will say, I was never there. So then you say, well, where were you? And so if you tell you, and you can test it, and they're never the same. So you have to take quite a lot of samples.
Okay. The third question, I find this really interesting. Can fungal spores be linked to the time of death?
But no, not so much just the spores, but you can do quite a lot if they're actually growing. And we did publish a little review of this, in fact, earlier this year, actually, in a journal called Expert Witness. This can be actually very, very valuable, but that's a separate talk. But basically, we know quite a lot about the growth rate of particular common spoilage fungi. The sort of fungi that grow on decaying bodies are those that will occur on decaying food and meat. They're not special things. They tend to be widespread saprobic things. So there's a lot of literature to do with food spoilage, particularly about rates of growth of these things. And so if you know what the fungus is and you know how you can measure your colonies, you can get an idea of how long that might have taken to grow. So it may have stopped growing, but you'll get a minimum amount of the time that's taken. And what we'd like to do, where possible, is to actually isolate the fungi as well and grow them artificially on a medium or substrate as close to the conditions in which it was found, so you can measure the actual rates of material that came from a scene. And if you do that, you don't even need to know what the fungus is. And again, with one case involving that technique that we did, we got something to the right day when somebody confessed, which was included a rapidly growing mucor, that one, and some two different penicillium species. So that it can be very helpful, and I've got a case like that actually that came in about 2 weeks ago with mold growth associated with the body, and they want to know how long that might have taken to develop. So you can do a lot.
I would say that in Britain, from my experience, a body doesn't go moldy for at least a week. So you always have to add on 1 week. Now, it might be different in India.
Yeah.
Because you have different climatic regimes and so on. So, but you have to do your own tests, really.
I mean, the human body obviously has resistance to fungi growing on it normally, otherwise we'd all be covered in molds.
Yeah.
And so there's a lag between where the skin sort of adjusts to when fungi can start.
Yeah.
And,
but please don't do that on animals, because it won't be. Don't start killing animals to test it. The best thing to do is to work with the police and do it from experience with crimes. And that's how you get a big database. We have a big database of knowledge from doing the actual work, not experimental. Experimental work isn't so valuable.
I mean, there was a case we were involved in where some people disputed some of the data we'd got to do with the growth rate of a fungus on a body, and they actually used pig. They've been using pigs as a model to try and grow a fungus.
Okay.
And I mean, there was various reasons why we destroyed those experiments in court legally. because the structures are quite different. The skin structures are quite different. You can't assume that anything will grow the same rate on a pig skin as it will on a human skin. If you looked at in microscopic section, they have different layers.
And of course, in America, there are 6 facilities called body farms. We are hoping to get one in Britain. They've got one in Canada. They've got one in Australia. And people donate their bodies when they die. And scientists are allowed to see what happens in the decomposition process. And in that, you can look at fungi growing on the body in those situations.
My personal question to Professor Hawksworth is, as an expert, how difficult was it, or was there any difficulty in convincing the legal body or people in your country for using fungi as pointers of some crime?
I've already done that, sorry.
Well, the legal body. I mean, basically, if you're a scientist, you can be accepted as an expert scientist. You don't need to be checked by anybody else. It's helpful if you are a fellow of the Chartered Society of Forensic Science or the Royal Society for Biology or so on, as I am. But basically, a judge decides whether you're an appropriate expert witness.
Okay.
And that's the ultimate thing. And so, we've had this tested on a number of occasions, and the judge will look at your CV and so on and say this person seems to be an expert. And of course, if you have a track record of appearing in court and things in Britain, there's a thing called the National Crime Agency that lists experts.
I didn't mean that, but
sorry.
Since this is something, let's say, new to science, but the legal body readily accepted. Maybe they are using Poland already, so probably you got to. They readily accepted this method of.
Well, it's been accepted because it's proved to be valuable in so many cases.
Mm-hmm.
The track record is there. Professor Wiltshire has been involved in not sure how many, probably around 300 cases now. She has 4 murders she's got on at the moment.
Excellent.
And so just the record is there, and so that's the evidence.
Mm-hmm.
And so the judges accept it. The judges are sort of all powerful in these things in Britain. I don't know how it works in India.
Yeah, exactly. That's what probably it varies with country.
Yeah, so the police don't decide what's good. At the end of the day, the judge decides.
Yeah. So, if there is time, there is one last question. Can you share an experience of yours where you faced the difficulty, and then but you were able to succeed using fungal spore as a pointer or something? Apart from what you mentioned in your talk.
Well, there are many cases where our evidence, the pollen and the fungi, have been the only evidence.
Oh,
so
okay.
No other evidence, no other forensic evidence. And it's been accepted by the courts and there have been convictions. But you have to plan what you're doing right at the crime scene. You must plug all the holes in a case. Yeah. And don't go into court until you're absolutely confident.
Mm-hmm.
It's quite fascinating when you're looking at these things, because you find fungus associated with a particular plant, and then you find another one associated with a different plant, but one that might be in the same habitat. Yeah. So gradually a picture of the place develops, as it does with the pollen. You get an idea of some of the plants that were around there, and so some indication of the place.
Yeah. So that ends this question answer session. Thank you, Professor David Hawksworth, for your interesting talk and your very convincing answers to the questions. Thank you very much.
Thank you. Sorry.
That's it.
I'm not sure what happens.
I think you switch it off. Yeah.
No,
You're trying to speak yourself.
Hello friends, I'm back again to express my gratitude through my vote of thanks. Sir Isaac Newton has rightly said, I quote, "If I have seen further, it is by standing on the shoulders of giants." Unquote. Yes, Professor David Hawksworth, a giant in the field of botany, has helped us see further through his talk. Sir, thanks for the very informative and lively session. It has helped us refurbish our knowledge about some rare fungal spores and their use in forensic applications. Truly, sir, your devotion and dedication in the field of teaching and research will serve as inspiration to the budding researchers. We are thankful to the senior mycologists and executive members of the Mycological Society of India for their support and the timely, valuable guidance which they render. Thanks to the principal and management of Satish Pradhan Nyan Sadhana College for their support and encouragement in the various endeavors we undertake. I am thankful to the entire organizing committee for their skills and expertise in the smooth and efficient conduct of the webinar. Sir Louis Pasteur has rightly said, and I quote, "Science knows no country because knowledge belongs to humanity and is the torch which illuminates the world." Unquote. Last but not the least, as thanks to all the participants who have joined from various countries all over the world in glorifying this occasion and making it a huge success. Thanks once again to all. A request to all participants to fill in their feedback forms, for which the link will be provided in the chat box and your respective WhatsApp and Telegram groups. Thank you, all of you.